it's composed. Riitta Lahesmaa is a professor, the head of the Turku Bioscience, and let's wait what she has to tell. Please.
Thank you, Sirpa. Thank you, Sirpa. And welcome everyone to this first InFLAMES Corporate Corner, also from my behalf. It's really great, exciting to be here and have you all here participating. So I'm going to talk about research we do on regulation of immune response. My team works in this building in Turku Bioscience, which is on the 4th, 5th, and 7th floor of this building, and very closely with Medicity laboratories that are on the 4th floor as well. Immune response is central for health, and it's composed of innate immunity and adaptive immunity. Innate immunity is the first-line defense, and adaptive is more trained, very sophisticated, and very tightly regulated. In adaptive immunity, we have many components, and one of the key orchestrator or the key orchestrator of adaptive immune response are the T cells. And T cells are the ones that recognize self from foreign antigens. And that's really crucial for health. When this fails, we are in trouble. So these T cells, T cells are naive when they are formed, and they, in the lymph node, they recognize the foreign antigens or self-antigens presented by antigen-presenting cells that can be macrophages or dendritic cells. And upon activation, and in the presence of, or upon activation, they start to divide, they proliferate. And then depending on the strength of this signal, TCR signal, as well as other stimulatory molecules, they are co-stimulatory molecules, cytokine milieu, they start also to differentiate. And they take different functionally distinct phenotypes. And this makes them able to really take care of their very, very multiple different tasks in our body. For instance, they can become, they get receptors that help them to migrate to the sites of inflammation where that is needed. And we will hear more about that later from Marko, for instance. And then once they have become differentiated, they then mediate their effects to other immune system by cytokines and other molecules they secrete, and also by cell-cell contacts. And then they can also take different shapes, different phenotypes, even later on. And all this is very, very tightly regulated, and it's important it goes right. We also have regulatory T cells, whose job is once they have taken, the effector cells have taken care of their job, they have to calm down. And we don't have ongoing effector cell function in our bodies. And that's why it's crucial that these regulatory T cells that have been educated in the thymus to keep this self-tolerance also to our own structures are properly working. And in a very simplified way, we can say that it's about balance. So you want to have a balance between effector cells and regulatory T cells. And when everything is in balance, they are interacting properly. However, when there is too much of effector T cell activity, we get autoimmune diseases, and there are too little of regulatory T cell functions. And how to take care of this is either you could try to stop these from working, or you could increase regulatory T cell functions by molecules, small molecules, or for instance, regulatory T cell therapies that have been developed by some of the very successful companies recently. And in cancer, the case is opposite. There you have too much of regulation and those effector cells cannot attack the cancer. And here you could either try to push the effector cells, or you could try to impair regulatory T cell functions, either directly functioning, trying to influence them, or by influencing the environment, such as macrophages that we are going to hear later from Maya about. So we want this balance. And our group is interested in how this balance can be achieved and what is behind this balance. And we want to understand in depth how human immune system is regulated. And through this, we hope to be able to discover new molecules and new approaches that help us to take this, to achieve this balance. And all this is to once we understand it, it's possible to diagnose and treat human immune-mediated diseases. So these events can be mimicked in vitro. And we have decided to focus on human T-cell activation and differentiation. There are many excellent groups working on the mouse system, and lots of basic information comes from the mouse studies. Why human? Because we treat human diseases, and I will give you a glimpse about that a bit later. But anyways, we use naive human CD4 cells that we isolate from umbilical cord blood. And we have really optimized this system to a very robust system so that we can really, we have created data on these cells, which we induce to differentiate to these different to different subsets, functional distinct subset, and we have collected 10 different time points during the first 3 days of this differentiation, because that's the state when these cells can be still modulated. You can change their fate if you want to. And this represents a type of time series that we have. And we have then used a range of different technologies and platforms to really carefully analyze these cells during their differentiation. And we have used different omics technologies. Importantly, we have also set up with Alex Marso's advice CRISPR-Cas editing for human primary T cells, so that we can, for instance, we have, depleted PIM kinases 1, 2, and 3, which now enables us to look how these T cells behave after these kinases has been depleted. And what is very important is computational analysis, and today we'll do a lot of collaboration with Laura Elo's group. So just an example, how do we use this? Here is an example where we wanted to look what is different between if you become a regulatory T cell or opposite effector cells, which is Th17 cell, which is important and harmful in many inflammatory diseases. So when we did this profiling, we wanted to look what are the most differently regulated during this time course of regulatory versus Th17 cells. And that was the most differentially opposite way regulated, and it's zoomed in here. And what you can see here, it's a huge upregulation of this zinc finger transcription factor in regulatory T cells, and it's downregulated compared to proliferating cells controls in Th17 cells. And what we found out was that this was a growth regulatory tumor repressor gene, zinc finger protein. We had not paid any attention before that. It's been very much studied in cancer, where its hypermethylation or deletion is associated with tumors. But what we identified it is as a novel regulator of regulatory T cell function and differentiation. And interestingly, it did not influence FOXP3, which is a known key transcription factor driving regulatory T cell functions. And very exciting finding was that when we did chip sequencing to identify where does this high C1 bind to, those sites had very high enrichments of SNPs that have been associated with immune-mediated autoimmune diseases, suggesting, and we showed that for some of them, that they can also change the binding of this transcription factor to this site. And now very new data has been obtained by Kedar and Bilal in our group, where they have made an interactome of HIC1. And I think that through that, we now actually understand how it works and how it, in a complex, it regulates this key differentiation process. So another way to use this is that with Gitta Stockinger, who invented or who discovered Th17 cells in the mouse in the first place, with her, we standardized and harmonized timescale of all these 10 time points between human and mouse, and did RNA sequencing to see really what's similar, what's different. And what we saw is that here we have the Th17 specification transcriptome between mouse and human, and there are shared transcripts that behave in a similar way, but there are also many that do not. And this is the shared transcriptome, and here is the human-specific transcriptome. These are not seen in the mouse system, indicating that it's very important when you go from human studies to mouse, is first check up how is your gene of interest behaving in the mouse. And we thought that, okay, this was transcriptome, but proteins are much more conserved, so we did proteomics, and it was even worse, to our great surprise. So, and there were among these such that were actually regulated in completely opposite way in human and mouse, and also functionally behaved in an opposite way. Third example, how we use this time scale is here, and this got me so excited, I must say, and the whole group, because it was first time we did RNA sequencing, and now we started to see something we never saw with the microarrays. And on the right-hand side there, you can see how genes are up and down regulated during time course, up and down of these first 3 days. These are protein coding genes. What is on the left-hand side? It looks very similar, the shape of the regulation of these genes. And they are non-coding, long non-coding RNAs, and nothing basically was known about them. And still very little is known about what they actually do. And of course, this became possible with RNA sequencing. And here is one of them where we have looked what does it do. It's we called it link RNA MK1, and you can see it's rapidly upregulated, much more than if you just cells just become proliferating. It's have very interesting localization, nuclear localization. When you deplete it, cells cannot anymore differentiate properly to the Th17 direction. When you look at its targets, there are quite many of targets, and when you look what are these targets, they are enriched in IL-17 pathways. We then looked at the targets and we overlaid with the kinetics of this gene expression during this early differentiation, and one of the most upstream upregulated targets was PKC alpha. And PKC alpha in turn has been previously linked to Th17 differentiation. So we think this is at least one way how this link MK1 regulates Th17 differentiation. And then this is not conserved in the mouse. It's not behaving the same way in the mouse. So how do we know, does it have any relevance? So we got our hands on this published data set, 2 sets, which are all single cell sequencing data from synovial biopsies of patients that have rheumatoid arthritis. And with Laura Elo's group, we analyzed this data, and who is there? It's our link MK1 that is upregulated in T cells, not in other cells of the synovial biopsies. And here you can see all the other genes out of genome-wide analysis. It's these ones that are co-expressed, upregulated with MK1. And if those who are familiar with immunology recognize quite many of them as very important factors, genes for T cell functions. So just to summarize this, so we identified a link RNA that regulates Th17 differentiation. And in fact, we have a few now also with functional data that also regulate regulatory T cell functions. So here are some of the selected papers from our lab. So if you get interested, have a look at them, and we are very happy to discuss more about them in the future. So I think I have a little bit more time, and I will then use that rest of the time to give you a glimpse about translational part of our studies. And we have a biomarker discovery of our studies. We have a long-term interest in type 1 diabetes, and that came when I long ago came back to Turku and started to discuss with Olli Simel at the time, and thought that this is really a very unique patient cohort we have here in Finland, which is the DIP study where they follow up children from the birth and then until they get type 1 diabetes. And in Finland, we have record high incidence of type 1 diabetes. And once you are diagnosed, you have to use insulin for the rest of your life. The autoantibodies are now used as indicators of ongoing disease. However, once you have autoantibodies, it means that immune response must have been started already. So we have been interested in looking into very early stages of this development of this disease. We know that HLA is important and genetic predisposition, but it's clear that there are very clear role for environmental factors. What they precisely are in each patient, we don't yet know, but there are these very strong hypotheses. So quite some time ago already, we worked collaborated with DIP and used different approaches where we identified signs of inflammation already before appearance of those autoantibodies. And in fact, Essi Laajala will defend her thesis tomorrow. She was a key player in these studies. However, these were whole blood samples, so all the cells mixed together. So it was obvious that we would like to look into more precisely the specific cell types, and we now were able to take part of this DIPE immune study coordinated by Mikael Kniep, and that was international study testing hygiene hypothesis. Now, what we were able to get were PBMC, meaning that frozen live cells. And now we had a very homogeneous group where we could really well match cases and controls, and most importantly, we could purify cell populations. And we published that transcriptomic signature a couple of years ago, and now in press is the methylome, the epigenetic signature of these children. And what was very clear from here is that when you fractionated the cells, you can see that this DNA methylation is very cell-specific. And another key point was that lots of these methylation events were seen already before seroconversion. And these are differentially methylated sites, which could be very important if you think about the environmental factors that might start to influence immune system in these children. So just to summarize this, this part has been already published. It was identification of gene signature before appearance of autoantibodies, and this second part will soon be published in Diabetologia. And in the end, I want to give a little teaser because this is something we are very excited about. And this is a technology, mass cytometry, that we have up and running here in Turku. It was developed quite some time ago by Gary Nolan at Stanford and has been used extensively by many people in both in UCSF and Stanford University, Mark Davis in particular, in profiling patients with, say, cancer or cancer immunotherapy or different inflammatory diseases. And it is now up and running also here in Turku. Maija Holmen has used it in her studies. Pia Rantakari has excelled it, and Marko Salmi looking into mouse responses. And we recently now, in our group, together with DIP and particularly Johanna Lempainen, has started to look at these children's PBMC in DIP study. And this is the panel we have. So we have used the basic panel of markers, human markers, but then we have enriched it with those that we are particularly interested in that are linked to these regulatory T cell functions. And this is just very first data. So Inna has done 206 samples run with this machine and done the basic analysis. There is a software provided by the vendor that can be used. Inna has used it herself. And these are the different kind of age groups that we have here. We have before seroconversion and before diagnosis. And what you can see, this is kind of giving you an idea, a teaser, that you can think about your own studies. So we can, Inna, would it be quite quickly to analyze this data with all these 40 markers and put your data into the context. And for instance, here is what you can see how CD4 T cells that we study a lot, how do they behave during these different time points. And we are now with Laura Elo's group developing a much more granular way to do the in details, the subsets. This is very broad way or superficial way, but it is evolving very, very well now, the data analysis pipeline. So with that, I hope you find our work interesting, and you are welcome to come to discuss with us if we can help you somehow in your studies. I would like to thank my amazing group members. They are really very, very excellent to work with, and they have done all this work. We have excellent core facilities in Turku Bioscience and core personnel. Without them, I think InFLAMES is strong because of these facilities and their personnel. And then we have super good, wonderful collaborators, enthusiastic collaborators. And perhaps I particularly point out here Laura Elo, because the type of approaches we have chosen really need heavy computational biology. Also, Harri Lähdesmäki has been important, and Essi, who defends her thesis tomorrow, is co-supervised by Harri and me. And of course, thank you for all the funding agencies. And with that, thank you, and wishing you a good holiday season.
Thank you very much, Riitta. Now, if there is a burning question, we will take that, but we are actually now use the time allocated. Is there any somewhere? So I have to say that this was a really great example that we are not mice and mice are not men, because that is so often seen in the drug development, that things are based on the mouse studies, experimental studies, and then they don't work in clinical trials. But people start to forget it.
Yes, something I wanted to say still is because you talked about these iPhDs, so I would like to welcome Anna Brunila, who is a new iPhD in our group, who is co-supervised by Anil Totakura and Anu Autio. So welcome, Anna.